All right, the next biological molecule we're going to look at is nucleic acids. Nucleic acids are made up of nucleotides. A nucleotide is a molecule with three parts: a 5-carbon sugar, either ribose or deoxyribose, one or more phosphate groups, and a nitrogen-containing base, which could have a single or double ring. With a single ring, we're looking at bases like thymine and uracil and cytosine, which are actually depicted just by their first letter, T and U and C. Or a double ring like adenine and guanine for the base, which are depicted again by just a single letter, A or G. Here's what a basic nucleotide looks like. Now this is a deoxyribose nucleotide, so its sugar is deoxyribose. This is what you would find in DNA. But regardless if we're looking at DNA or RNA, their nucleotides are made up of a phosphate group, a sugar, and a nitrogen-containing base. These repeating units, when covalently bonded together, form nucleic acids. And nucleotides by themselves can be energy carrier molecules, intracellular messenger molecules, and even subunits of nucleic acids. Differing types of nucleotides do exist based on changing out those bases. One example of an energy carrier that has a base structure that looks like a nucleotide is adenosine triphosphate, or ATP. It has ribose sugar and a phosphate group, and adenine as a nitrogen-containing base. And it's known as the energy currency for cell. It's able to break down phosphates of its 3 that it has to release energy, and that's actually going to help with the breakdown of sugar and metabolism in many organisms. So here's that structure for ATP. You can see it has a green nitrogen-containing base, that's adenine. It has a ribose sugar, and then it has 3 phosphate groups. So it's a little different from our normal nucleotide. Those last 2 phosphate groups, bonds between them can be broken off to release energy to power parts of cell functions. And ATP is able to store energy in its bonds, and then when those bonds break, it releases those energies. So you can both build and break down ATP. This energy really drives many of the different types of reactions within organisms. But let's take a closer look at nucleic acids. These are what are made up of many repeating units of nucleotides. DNA and RNA are 2 examples. They're the molecules of heredity, and RNA tends to help out DNA in deciphering the genetic instructions. So nucleic acids are just polymers of nucleotides. They're really long, linked together chains of nucleotides, and they're formed just like carbohydrates and proteins by dehydration synthesis. And the oxygen in the phosphate group of one nucleotide will be covalently bonded to the sugar of another. So it's the phosphates and the sugar that make up the backbone of DNA and RNA. The bases that are left over, they're going to make up the rungs of what looks like a ladder for DNA and RNA. So let's look at DNA first. DNA has deoxyribose sugar, which is why it's known as deoxyribonucleic acid, and we just abbreviate that DNA. And it contains millions of nucleotides. And these nucleotides are in an order that provides the genetic instructions for how to make proteins. It's actually the sequence of deoxyribose nucleotides that spells out the genetic information for how to make a certain protein. In what order should we put amino acids together? DNA is double-stranded. On the outside, its backbone. It has sugar and phosphate groups, and its bases in the middle are connected via hydrogen bonds. When it comes to the bases you find in DNA, they include guanine, cytosine, adenine, and thymine. Adenine and thymine always pair together, and cytosine and guanine always pair together. How can you remember that? Well, you want to put the car in the garage, and apples are found on a tree. That's one way to remember A pairs with T, and G pairs with C. So let's review. DNA is a nucleic acid that is double-stranded. It's the genetic blueprint for how to make proteins, so it dictates protein synthesis. It also contains bases A, G, T, and C. RNA, on the other hand, has ribose as a sugar, and it's single-stranded. You can find ribonucleic acid, or RNA, in the nucleus and in the cytoplasm. So it can talk to DNA and then leave and talk to ribosomes, which are structures in cells that make protein. So it's really a go-between molecule. It's copied from DNA, and it directs protein synthesis. So it uses instructions or information it gets from DNA to do that. So to review for RNA, it's single-stranded, copied from DNA, and there's 3 types that help synthesize proteins directly. Here's a quick view of RNA versus DNA. You can see the different bases that are in DNA versus RNA. RNA on the left, which is single-stranded, has uracil instead of thymine. Otherwise, the bases are the same for RNA and DNA. We don't have a second strand, so you kind of have an unfinished ladder for RNA in that single-stranded view on the left, whereas DNA with its double strands uses its bases as kind of rungs on a ladder, and they're all base paired together. Next, we're going to be looking at lipids, so they're going to be one of our last biological molecules to look at, so we can finish up our look at organic molecules and their polymers.